Mm, Okay, I think I have admitted everyone. Hi, everybody. For those who don't know, my name is Buriana Rosa, and I'm an artist. I'm a professor in transmedia department in Syracuse University, and together with Professor Heidi Henley, we have been doing BioArt Mixer for at least a couple of years, I think. Yes, this is our first online BioArt Mixer, which is really altering our protocol. Very important part of it was the hanging out together and being able to talk to each other informally, which I'm not sure we can really condition in Zoom, except in a private chat. Uh, so it will be definitely a very different format. But still, we decided that it is important to have it again in this weird situation, just because we want to talk about something else, not only the virus.
So I'm going to start. I'm Heidi Henley. I'm the biology faculty. That is my dog and probably a child you're hearing in the background. I'm sorry. I'm super excited to introduce Maggie McCoy, and I'm really happy that she agreed to give a talk today. Maggie is a master's student in Melissa Pepling's lab, and she's going to be talking about the effects of anesthesia on germ cells, and probably some other cool stuff in there. When she was talking about it, it sounded really exciting. So I think it will fit really nice with our second speaker today, which I think Boriana is going to introduce to you.
Yeah, I want to introduce Kira Riley, who is an amazing artist who has a practice in so many different fields, such as performance art and photography and text and bioart. And I think this combination of activities and mediums is what is making her very precious for our meeting today. Because a lot of, I think this is something that we haven't really presented so far, the connection between the human body and biological research, except in Kati Hai's presentation. So far it was mostly artists who are dealing with material, with biological material using biological protocols, but this direct connection between the human body, human emotions, human experience was present mostly in Kati Hai's presentation. And now we'll have Kira O'Reilly, who extend this kind of an aspect, at least this is how I understand her presentation, but maybe it will be something different. But the understanding of bioart as something that can include also thinking about the human body as a material, an object, subject, both emotional and physical. Kira has been, she is now a professor at Alfred University. I think he's a visiting professor. And I don't know exactly your title there.
I'm the, what am I? Randall International Chair in sculpture.
Okay, so in sculpture. And before that, she was the head of a new program in the Helsinki Art Academy, which was focused on performance art and ecology. And before that, she had, and she still has this huge career of an international artist presenting her performances internationally, acclaimed by media and by different artistic institutions all over the world. So I'm really happy to have her here, but I think we'll be starting with Margaret, and Kira will be the next.
Okay, should I start?
Yeah, you can share your screen. I think I gave this permission, so we'll be watching your screen.
Okay. All right, let me. Okay, everybody can see my screen?
Yes.
Okay. Hi, I'm Margaret McCoy. I typically go by Maggie, so if you want to refer to me in the chat as Maggie, I will respond to you. I am a master's student in Dr. Melissa Pepling's lab, and today I'm going to be talking about the effect of general anesthesia on germ cells. So this first slide that I have up, these are a mouse ovary, a confocal image. So what I'm highlighting with my cursor is a mouse oocyte, or what you could call an egg cell. So this is what could develop into another litter of mice. So to begin, I would like to talk about the broad history of reproductive toxins. When we think about a reproductive toxin, we can think of something that will affect the adult reproductive organs of a human, so that being a testis or an ovary. But for the purpose of this talk, I'm going to be talking about reproductive toxicity and the effect on a developing embryo or the fetus. So I'm really thinking about that embryo that's growing in the uterine environment. So one example of a reproductive toxin would be diethylstilbestrol, or DES. This was a medication that was given to pregnant women between the 1940s and 1980s in hopes of preventing miscarriages and early births. And while DES did this somewhat, it's actually a reproductive toxin. So females that were in utero while their mother took DES actually have a 40% chance of developing clear cell adenocarcinoma and in their genital tract, which is a type of cancer, and they have other adverse health effects. So while this might have somewhat helped to stop miscarriages, to prevent miscarriages, it also was a reproductive toxin. Another famous example of this is thalidomide. Thalidomide was a drug that was used in the late 1950s to just the very early 1960s, mainly in Europe. Thalidomide was prescribed to help with morning sickness, but in reality, it's a pretty horrible teratogen, which means that it's going to affect development and cause deformities. This is also because it affects angiogenesis, which is the development of new blood vessels. So if a female is pregnant and taking thalidomide, it might actually stop those new blood vessels from forming, so it might cause a hand deformity or foot deformity. And it very much depends on the window of when the mother took the drug. So if it was early on, it could have more severe deformities than if it was taken later on in the pregnancy. So these are just ideas of reproductive toxins that I want to think about, and then also think about how the uterine environment reacts to toxins. And sometimes it's not, sometimes it's you can't avoid certain toxins. So with that idea, I kind of want to talk about anesthesia, because anesthesia is something that a pregnant woman might have to undergo, and it can't be avoided. So approximately 2% of pregnant women in the United States will undergo anesthesia for an issue unrelated to gestation. Women who undergo anesthesia are more likely to deliver preterm, and their offspring typically have a lower birth weight. This can be correlated with the fact that they delivered preterm. It would make sense that the offspring might have a lower birth weight. And sometimes this is also due to the underlying condition of why they underwent anesthesia. So it might not be the anesthesia itself, but it's hard to parse that out. So thinking about anesthetics and that they might be you can't avoid them. In the 1950s to the 1980s, halothan anesthesia was brought into the scene of general anesthetics. It's a very volatile anesthetic, and it depresses the central nervous system. So this is how it anesthetizes a person. When it was first introduced onto the market, there were about 2 cases of severe liver hepatitis. And this is because when halothane is metabolized by the body, it produces free radicals, and this can lead to liver peroxidation. So it's pretty toxic to the liver. Although this didn't happen in most cases, and most people who who had halothane anesthesia were able to have normal liver functioning. When we think about toxicology and anesthetics such as halothane, we need to think about how they are absorbed by the body, and then they undergo disposition, and then how they're eliminated. So for a toxicant such as a gas like halothane, the elimination of the gas is inversely proportional to the rate of absorption. So if the toxicant is absorbed really well through the lungs, that means it's going to be harder to eliminate. So when we're thinking about halothane is very lipid soluble. So that means it's really soluble in the blood. So it's going to be absorbed into the body much better than other anesthetics. And because of this, trace concentrations of halothane can actually be found in expired air when an individual breathes out for as long as 2 or 3 weeks after when the individual had the anesthetic. This is because the halothane is being sequestered in the adipose tissue or fat tissue. So fats are made up of lots of lipids. So the halothane, when it's readily absorbed into the blood, can be stored within the fat tissue. And that's why you can find it in the expired air for a long time after. So when we're thinking about these anesthetics and different toxins, it's interesting to think about how that could affect a developing embryo or a fetus. Most general anesthetics can cross the placenta through simple diffusion. So that's kind of what I want to keep in mind. So when we're thinking about how anesthetic like halothane could be toxic on a developing embryo, I kind of want to draw our attention to epigenetics, because this could be one way that it's causing toxicity. So what is epigenetics? For those of us that aren't familiar, the epigenome is a collection of different chemical compounds that are going to kind of control the genome. So most cells in our body have the same DNA, so that underlying alphabet DNA sequence. Your eye cell and your skin cell probably have the same genome within their nucleus. But why are those cells so differentiated? Part of that is because of the epigenome. The epigenome is going to be this regulatory network that is going to interact with the DNA and control gene expression. So epigenetics won't actually change the physical DNA sequence, what you inherited from both of your biological parents, but it is going to change how those genes are expressed. So when we're thinking of that, epigenetics can cause a lot of different regulatory problems in the body. So for the purpose of this talk, I want to talk about DNA methylation. And there are a lot of different epigenetic mechanisms within the body, but I'm just going to focus on methylation for the purpose of this talk. So what is DNA methylation? So this is essentially a methylated cytosine. So it's a methyl group that's going to attach to a cytosine nucleotide. So it's going to be this little chemical that comes in and attaches to that cytosine. And what this does is it's going to kind of like mark that cytosine. So now there's a physical chemical tag on it. And when that happens, if that happens in a gene promoter region, it can actually block that gene expression. In a gene promoter region, there are, when that gene needs to be expressed, there are different proteins that are going to come in and connect to that promoter region and cause the gene transcription, and then the transcription leads to gene expression. But if you have that methyl tag there, those proteins that interact with the promoter region can't get in there, and so they can't cause transcription, and therefore there won't be any gene expression. So methylation is a really important part of epigenetics and of gene regulation. So when we're thinking about epigenetics, it's important to know that the epigenome is dynamic. So it's constantly changing. It's not static like it's not static like DNA. So we know that parents have a big influence. So the epigenome is dynamic, so it's constantly changing. So when we think of this, it changes most often because of our environments. So our first environment we kind of interact with is our parents. So we can think about parental influence in this. And we know that parents have a huge impact on the cognitive and the emotional development of children, but they also might have an impact on our epigenetics. So when you're in utero, this is your first environment, your first epigenetic environment that you're going to have. While you're developing as a fetus, your epigenome is changing. So how your mom interacts in that pregnancy and what toxicants she's exposed to could affect your epigenome. And here we can kind of see how over the years I mimicked my mom's attributes between standing and my head tilt, and who knows where that came from. So when we're thinking about the epigenome, like I said, it's dynamic and it changes with our environments. So if you looked at the epigenetic profile of two monozygotic twins, so identical twins, these twins have the same genome. When you look at their methylation profiles later in life, it's very different depending on how close these twins stayed to each other. This is because they probably had different lifestyle choices, smoking, drinking, exercising, diet, and just different environments. So the first environment we're in is within our mother's uterus. So when we're thinking about this, it's like, all right, we inherit our DNA from our parents, but if the epigenome is constantly changing because of our environment, we wouldn't want to inherit that, right? I wouldn't want to inherit my mom's epigenome because of some trip or something, right? So you want to have a fresh start with your own epigenome. Because of this, when a zygote becomes fertilized, so the egg and the sperm, and you have a zygote, it undergoes rapid demethylation. So those chemical mark tags that change the gene expression, those are kind of erased for this new developing embryo. So you can see here on the chart, you can see this is the percent of methylation, and then you're going to see this rapid decrease here, right? And then the embryo is going to undergo de novo methylation, so new methylation for that own embryo. And so this is when you need a heart cell specialized. That's going to happen at this time. The methylation is going to, specialize that cell so it can vibrate. At the same time, there's these primordial germ cells. I'm just going to call them germ cells too. And these primordial germ cells are what's going to develop into a reproductive organ, so a testy or an ovary. So you can think of germ cells as the gonads, as the reproductive organs. So these primordial germ cells are set aside at around 2 weeks in humans and embryonic day 6.5 in mice, 6.25 in mice. And then these primordial germ cells, they undergo methylation erasure again. So we can see right here, these migratory PGCs undergo methylation erasure. And this is because you wouldn't want your ovary or your sperm to again have different methylation. You want it to have a clean methylation just from those germ cells, or be able to undergo its own de novo methylation. For males, it does undergo de novo methylation in mice at about embryonic day 6.5, so after sex determination. But for females, it remains at a low level of methylation until meiosis II is resumed. So female primordial germ cells are stuck in diploatine of meiosis I, and they don't actually become remethylated fully until they enter meiosis II, which is typically when they're fertilized. So it's really interesting to think about how this methylation occurs and what affects it. So with thinking about this environment and different toxicants and how methylation occurs, I want to talk about like intergenerational exposure, because this is what my project is basically based on. So I don't know if I should move this. Okay. So when we're thinking about intergenerational exposure, we're thinking about 3 generations. So like I said, we have the gestating mother, and this is going to be that F0 generation. And then we have the fetus, or the developing embryo, and that's going to be the F1 generation. And then we have the germ cells, or this F2 generation. And that's really what I'm interested in. So we know that each of these individuals is going to have their own epigenome. But what's interesting is that while they're all in this environment together, the gestating mother, the developing fetus, and then the F2 germ cells, how the environment interacts together can affect these F2 germ cells, because they're undergoing that methylation erasure and then that remethylation. So it's a really critical time point. So when we're talking about a toxicant affecting gestation, we're talking about this intergenerational 3 generations of exposure. And there are certain, there's anecdotal data that suggests that, I'm sorry, I moved the chat and it kind of threw me off. So when we're thinking about this exposure and epigenetics, it's interesting to think about different biological diseases and disease origins that might arise through irregular methylation and gene expression. So there's been kind of anecdotal data that might suggest that exposure to general anesthetics could cause some type of autistic-like phenotype, but this again is just kind of correlation epidemiological data. So we're kind of doing a base study here to see, and we got funding from the Escher Fund for Autism to do this work. So my broad project goal was to determine if in utero exposure to halothane anesthesia affects the methylation profile of germ cells, and then if this methylation could cause intergenerational cognition effects. So I'm thinking about if this population gets anesthesia, if it could affect this F2 germ cells. So when these individuals are born, the grandchildren, if they could have effects from that. So the first aim is to determine if this exposure causes cognitive and behavioral irregularities in the F1 and the F2 generation. Mainly, we're focusing on the F2 generation because they were those germ cells while in utero. And then the second aim is to assess what regions of the genomes are actually differentially methylated. So what specific genes or areas are going to be affected. And then we could maybe see later on how this affects the actual gene expression. So to do this, I exposed pregnant mice at embryonic day 14.5. So this is right kind of after sex differentiation, but it's when both the male germ cells and the female germ cells are at their lowest point of methylation erasure. So male germ cells have not begun to get that de novo methylation, and females are still at low levels. So this is a kind of a critical time point. I had 3 different testing groups. My behavioral testing group, the green box is the box that I'm going to do that methylation analysis on, and then a breeding group. The reason why I needed 3 separate groups is because, like I said, the epigenome's dynamic. So even in mice, behavior testing, being brought up on a cart, listening to people maybe talk, could actually affect their epigenome. If they get stressed during the behavior tests, or if they get really excited, that could potentially change the epigenome. So I wouldn't want those behavior mice to influence just what the anesthetic did. So I had those 2 separate groups, and then my group just for breeding of those grandchildren, or that F2 group. I exposed the mice to the halothane anesthesia for 30 minutes, starting at an induction of 3% and then a maintenance of 1.5%. So the first thing I'm going to talk about is that behavior testing and how I did that. For the purpose of this talk, I'm just going to share my F2 data, that grandchildren data. No data in my F1 generation was statistically significant. So there's not a ton to share there. And then for the purpose of this time limit, I'll just share the F2 data. So I'm focusing on, this was the behavior of the mice that were the germ cells while the anesthesia was administered. So the first test I did is called the elevated plus maze, and this evaluates mouse anxiety. So here you can see I just have a simple mouse behavior assay, and it has 2 open arms right here and then 2 closed arms. So a mouse that is more anxious typically spends more time in these closed arms, and a more exploratory mouse will spend more time on these open arms. Mice are very curious. If you stick your finger by them, they come up and smell it. So they should want to explore all arms of this test. And then we use a cool system called EcoVision, and it monitors them through heat software. So this is what my arena looks like and how I collect the data from this. So this is one of my first graphs of the elevated plus maze. So this is the F2 generation, and this is the maternal side. So mice, male mice that were born from this, their mother was exposed to halothane. So a 2A ANOVA of this data showed that nothing was statistically significant. The control mice that were only exposed to oxygen spent as much time in the closed arms and the open arms as the same as the halothane mice. For the female cohort that was maternal, so again, their mother got anesthesia, it was the same. Both animals spent relatively the same amount of time in each arms of the test. The control female spent a little more times in the open arms, but nothing was significant. Then for the paternal cohort, so their father was exposed to halothane anesthesia, there actually was a significance. So the halothane male mice spent more time in those closed arms than in the open arms. So they did show a little more anxious phenotype, and they like to stay nuzzled up in that closed arm of the test. So it does speak to some anxiety-like phenotype, although behavior data is somewhat subjective. And then I had the elevated plus test for the paternal cohort and females, and again, the data is the same. They spent the same amount of time in both arms of the test. The next assay that I ran was the social approach test. So this is going to assess sociability in mice. Mice, again, are really social creatures. They love to be housed with each other. They don't like to be in isolation. So this test will kind of see how social do they want to be. So here you can kind of see the arena that I have. there's 3 chambers within this arena. So there's a middle chamber where the mice become kind of accustomed to this. They get habituated. And then there is an object side. You can kind of think of this object. It looks like an upside down pencil cup. So it's just kind of like has little bars on the side. And then on the other side, there is the same little pencil cup, but then there's a mouse under it. The mouse under it is both age matched and sex matched. So there would be no like breeding effect or mating effect here. And then you want to see how much time the mouse spends either with the object or with the other mouse. If this works, I have a little video. So you can kind of see the mice are freely able to go between all of the chambers. This mouse is interacting with the other test mouse, and this mouse is interacting with the object. And it's very quiet and silent in the room. There is a fan going, but there's no outside noise, nothing external. So they're kind of just allowed to freely roam. So the data from this test for the maternal cohort, the males spent more time, all groups spent more time in the mouse chamber. But again, both groups spent about the same amount of time. For the females, again, same thing. Both groups spent significantly more time in the mouse chamber, although there was no difference between either. For the paternal cohort and males, again, they all spent more time in the mouse chamber, but there was no difference between the halothane group and the control group. And then for the paternal cohort for the females, there was a significance. The halothane females spent more time with the mouse than with the object. And this was kind of opposite of what I hypothesized. I thought that the control mice would spend more time in the mouse chamber than the halothane females did. So the second aim of my project was to assess what regions of the genome are differentially methylated. So to do this, I use reduced representation bisulfate sequencing. So what this does is it uses a restriction enzyme to cut out sections of the genome that are really just cytosines and guanines, which we call like CPG islands. And this is where methylation typically occurs. So it's a good method to use because it'll get us the areas of the genomes that are typically methylated. Then it'll undergo bisulfate conversion. So any cytosine that is methylated will stay a cytosine, but a cytosine that wasn't methylated is going to become uracil and then thymine. And so that's how we're going to be able to tell the difference between what was methylated and what wasn't. Some of you might have heard of whole genome bisulfate sequencing. It's very similar to this, but whole genome bisulfate sequencing sequences the whole genome, so it's very expensive. And because this is reduced representation, it looks for those CPG islands. It's a little bit more cost efficient, which is awesome. So I collected genomic DNA from the male sperm, and then I collected genomic DNA from the whole ovary. I have 2 biological replicates per condition. So that means I sampled 2 halothane females, 2 control females, 2 halothane males, and 2 control males. And then I had the sequencing done at the Cornell Epigenomics Core. You just get to see this cool, not so cool picture of my coding. I'm still analyzing this data right now in R, so I still have kind of quite a bit to go on this analysis, so I'm not sure yet, but we can see that these are my samples that I sent in, and ME means methylated, and this is unmethylated. So I'm gonna work and do some more analysis to see how the anesthesia actually affected the methylation of the genome, of the reproductive cells. So with that, I conclude my talk. These are some of the references I used. And I would really like to thank the whole Pepling lab. As you can see here in the picture, I started in Melissa's lab when I was, I think it was the summer of my sophomore year, but more of my junior year of undergrad. And then I've stayed on to do my master's in her lab. I'd also really like to thank Lindsay Banks. She's another master's student in Dr. Pepling's lab, and she's helped me a lot with some of the behavior testing. And Sue Getman, our lab manager, she helps me exponentially with the mouse colony and breeding and ear clippings and all the fun things that take a lot of time also. And Josh Burton, the other graduate student in our lab, for listening to me talk about this project a lot, Dr. Pepling, and then the members of my committee, and also members of the McDonald lab, Nico and Mai, who have helped me a lot with the behavior testing.
Thank you, Maggie. That was awesome. I think usually what we do is we go right into our artist, and then Ed will help promote discussion on this thing. So yeah.
That's what we usually do. Yeah, and it's always a kind of distinct challenge to try and find the overlap between the talks. So if that's something we can invite everybody to try and
And this is your challenge.
it's like a very perverse challenge, but I accept it. But I invite others to help me.
Yeah,
that was excellent. That was really interesting talk.
It will be interesting that these two presentations may be related to certain parts of the body which are the same. Approach is very different, and maybe they talk about something very different, but still it is about this part of the body which is related to reproduction, somehow present in both of the presentations. At least this is my understanding of what is common. Yeah. So Kira, I see you.
Hello, yes, here I am. Thank you, Maggie. That was fascinating. I wanted to spend much longer on each slide, but it was just because I thought, wow, there's so much here, and so much lovely detail, and so big thank you, and what a pleasure it is to be here. I really appreciated that very generous welcome, Oriana, and introduction, and it's lovely to see everybody. Lots of pages and lots of names.
We have 45 people here.
Yeah, I'm a little intimidated. So what I'm going to do is I'm going to read to you. There isn't going to be any sharing of any visual material apart from changing my backdrop from Neptune to a picture from a performance that I did about 3 years ago or something. And I've been presenting writings quite a bit over the last few years that are what I call performative writings. They're writings that are written by me to read to people in live situations. They're not really texts written to be read by someone else. So they have this characteristic of me performing them, which is just really me reading them. I'm not going to do any silly voices or anything, not in this one at least. And I've not done this online before, so it could be just, well, we'll see what it's going to be, but normally I'm relying on being in the same room as people, and the kind of atmosphere that we generate together as a community or as an assembly. So with a virtual assembly, I feel really curious, actually. I feel quite intrigued. There's nothing you have to do except listen, and whatever else you're interested in doing at the moment.
Kira, would you mind if I invited people to almost sort of free associate in the chat, so to sort of capture people's responses as we go, try and cultivate that idea of an assembly?
It's a gorgeous idea, and that's a really generative idea, which is kind of how I've been writing. I just make collages of text. So this is going to be quite kind of poetic and tangential and not very straightforward. So any sort of tangential writing, be it a solid sentence or just phrases or just kind of floaty things, would also be really gorgeous, and very, very welcome indeed. And that performance of generating text, and I've been interested in where, you know, where text and language, words and utterances become embodied, become the stuff, become material, really, become something solid, and where do those things then also become quite kind of abstract and just about science, just about semiotics, about something that's very, very removed and very academic and so on. And suddenly, as I'm saying that sentence to you, I'm suddenly hit by this absolute self-consciousness of feeling like I'm just talking to my kitchen wall or something. You know, I'm in my kitchen at the moment, or the kitchen that I'm renting. It's such an interesting feeling. So as Boriana mentioned, over the years, I trained in visual art, and I worked in the early part of my professional career pretty much exclusively with making performance art. And the logic of making performance art was really a sculptural endeavor. It was this idea of my physicality being a malleable, mutable material, something that could be moved and that is undergoing process, that doesn't have a sort of definitive final stage, but is always in some kind of flux. And then that brought me into working in bioart to some extent. And that expanded, I'm just going to give you a little sort of lineage in a way, that expanded into my kind of realization that the human body is only one category of liveliness, and that my thinking could dissolve the human body into lots of relations, lots of ideas, that the kind of fixed body, be it human or some other kind of species, is a moment in time, or many, many moments in time, but through those moments in time and through those gathering of process and materials, there could be many other questions, many other thoughts about what subjectivities might be, or what figures might be, or what bodies might be. And so that was really informed by the kind of abstractions I experienced in biology labs, in laboratories of life sciences, where I was encountering mice and encountering molecules and encountering all sorts of reagents and substances, this distribution, this abstraction and distribution of bodies and combinations of materials. And then a later stage came in my work and thinking, but through the benefit of being around a lot of other artists and thinkers and people and places and non-human others, which was then a further dissolve from thinking about human and non-human animal bodies into thinking much more environmentally. So there's another kind of suspension of a category, or questioning of category of body or animal into something much, much, much more expansive, which is environment. So I suppose where I'm at now is a kind of expansion and contraction, and thinking across scales and thinking across durations. So my poor students, since the beginning of this year up till now, have had to work with these two key words, one being duration and the other scale. And I've been inviting them to think about duration and scale in multiple ways simultaneously, and they're kind of over it now. They'll stop. But that's just to give you a little bit of background into where this writing is coming from. So for the last few years, I've been particularly looking at menopause. When I entered into perimenopause some years ago now, I should really work out how many years ago, I felt that there was a whole load of cultural and social expectations and constructions of what this is, this extraordinary transition, that I rejected, really. And maybe that's from many years of feminist inquiry through artistic practice into biomedical histories, thinking about how female experience can and has been so medicalized and told within very particular stories, I suppose, of experience. And I felt, that I felt resistant to the version that I was seeing my peers going through, which was just to get really, really fucking miserable, and to have another iteration of another curse. So I'm explaining this really badly, but that's a sort of short version. And I became really inspired, and I think remain inspired by wanting to position other embodiments and other kinds of journeys and stories. And this isn't to diminish anyone else or any of my very important and very special friends. It's just me being the contrarian, really, and wanting to delve and discover and uncover other possibilities in terms of my artistic thinking as well as my personal thinking. So that's a kind of preamble. That was a bit long as a preamble. So I'm just going to start reading. The printer didn't work today, so I'm going to read off the screen. And I'm actually going to change my virtual background as well. So we have some sea. I should go this way, really, and a little bit of me by the sea. And the other thing I should say is normally I read this all into a mic right in front of my mouth, so who knows? Sigh. Unseamed. Unseeming. Unseemly. Pause. Pause again. Pause some more. I fear that I'm going to be unseemly, indelicate, indecorous, unbefitting. That this seeming lack of composure is an undoing of sorts. Certainly it feels that way. An undoing of some version of whoever might be before you. I'm going to sigh again, not from loss of demeanor, but of queasiness, from trying to find breath, trying to locate a spell that will maintain what can appear like an all too delicate balance of pressures, of air, breath, and atmosphere. Lily, the caretaker's daughter, was literally rushed off her feet. Where was Lily? Where was her body? Performance studies scholar Peggy Phelan asked this question as she read the opening phrase from The Dead, a short story written by James Joyce in 1914. I was one of the participants at a 3-week summer school on the top floor studio of the Centre of Contemporary Art in Glasgow in June 1998. Peggy then asked us to write down lists of expressions that revealed the body in language, somatic hauntings in idiomatic phrases. By the skin of my teeth, I have my eye on you. I've got cold feet. I paid an arm and a leg. I'm all ears. I need to get it off my chest. I'll stick my neck out. I'm head over heels. They saw eye to eye. I'll play it by ear. It makes my blood boil. My lips are sealed over my dead body. Sweat, meteorology. Horses sweat, men perspire, women glow. I did not mind the arrival of the sudden night sweats that flashed like floods drenching me, despite their disruptive drama. Rather, I enjoyed their excessiveness and sense of abandon. I didn't mind being sodden. Somehow it was satisfying, intense, and tropical. Perhaps I found myself unbothered because I was used to sweating like a horse. There was a moment sometime prior to the onset of hormonal deluges when I announced that I, quote, sweated like a man, unquote, when I trained in martial arts. There was nothing restrained or glowing about my salty deluges. I poured unrelentingly until drenched. My face went red, my hair all over the place. I precipitated, I misted, and I formed clouds. At the end of each training session, as we stood in line, making to make our final os, we would pour, sweat, perspire, glow, forming a microclimate from our embraces and exertions. Humidities would ascend the uncladded railway arch roof, only to return in liquid state and pool on the mats as cold, briny puddles. In Seeing Gender, Kathy Acker wrote, "When I was a child, the only thing I wanted was to be a pirate. Because I wasn't a stupid child, I knew that I couldn't. I couldn't send men down the plank. I couldn't see sights stranger and more wonderful than those seen in my childhood dreams. I couldn't dwell in seas that would freeze my lips and whose living and dead denizens would tear away my bones. I couldn't swing from any yard arm because, I announced, my parents won't let me. If only my parents were dead, I could do all that I wanted to do. I could run away to sea." I am 53. For the last several years now, I've been exploring and writing about being environmentally menopausal. I've been thinking about how to reframe menopause and how to become a pirate. Menopause is turbulent. One needs sea legs that are sturdy and can move in accordance with changing weathers, fortunes, and seas. One is literally out at sea, uncharted sea over which the normal starry constellations are absent. There are stars, but they are arranged into hitherto unrecognizable orientations. The cardinal points have all shifted. Perhaps the poles have moved to the equator and Lapland has come to me. Menopause is a series of transitionary states during which hormones soak, saturate, and abate in exotic tides. One's very self is up for grabs, and there is no clear sense of anything. This pervasive nonsense is rich and strange ground in which things do not exactly grow, but emerge. In our endocrine altered environments, insides and outsides are subtly permeable. Phytohormones and animal hormones, biochemical molecules that are hormone-like, that masquerade and alter, all travel across and through bodies of plants and animals and ecological relations, as bodies are processes, and processes are transformations of substances and things. Menopause is environmental. Menopause is timely. It is about time. Sweat Protocol 1. Be menopausal, go to bed, throw off covers. Sweat Protocol 2. Hold a copper pipe closely in the heat of the day under the hot sun. Glisten. Sweat Protocol 3, version 1. Hold a cast iron kettlebell, perform swings with it energetically, sweat, salt, and water, causing the kettlebell to rust. Version 2. Hold a cast iron kettlebell, perform swings energetically, perspire clouds that precipitate, causing the kettlebell to rust. Sweat Protocol 4. Wear a white cotton mutated laboratory coat to the gym. Run and or row into a heavy sweat. Use the coat to absorb the sweat. Bury the sweaty coat in a wild area in the hope that sympathetic microbes will grow with, on, and from the sweat. I'm trying to build muscle mass. Pause. I read somewhere that it's the best thing to do for a woman of my age. Pause. Rather than head down the road of a gender-conforming regimen of hormone replacement therapy just yet, I'm trying to manipulate and work with my own physiology and hormonal well-being. Pause. The coach does not appear to like me to refer to menopause. Pause. Or that I am menopausal. Pause. And I am sure he entirely unintentionally treats me as more frail and with less capacity than I have. Pause. I try to explain that skillful coaching of perimenopausal and menopausal women is an untapped market ripe for the picking. Pause. That helping women architect muscle to support their well-being as they transition in, through, and beyond menopause and its post is a terrific idea. Pause. And that we make testosterone, that our muscle mass is intimately meshed with the delicate production of that culturally affirmative, biologically determinate elixir of androgen prescribing sovereignty. Pause. I feel a little condescended to. Pause. But not enough to put me off. Pause. He wants me to Instagram my visits to the gym, or now my home workout sessions. Pause. I don't want to, but perhaps if I hashtag menopause. Pause. I explained that I want to understand the muscular influence on metabolic operations and endocrine pathways, and so to calibrate my own hormonal profiles. Pause. The artist and physical trainer Castles tells me about Diane Nyad, the swimmer who was only able to realize her self-imposed challenge of swimming from Cuba to Florida in her early 60s. Pause.
Steak with roasted hazelnuts and onions.
I read more about muscle mass and muscle loss. The regulation of muscle mass is of interest to a diverse group of people. There are those, such as power athletes and bodybuilders, who are primarily interested in increasing their muscle mass. Others are concerned with preventing muscle mass. This is critical for the frail elderly, those with myopathies, cancer, sepsis, HIV/AIDS, and other diseases who suffer, and other diseases, those suffering from reduced mobility as a result of injury, and astronauts. Sorry, that was a quote, by the way. End of quote. Pause. Astronaut. Pause. Pausal-naut. Pause. Astropause. Pause. Today I found a copy of Getting Built for Women, ! A bodybuilding program for beauty, strength, and fitness by Lynn Pyrie, with, in much smaller writing, someone called Bill Reynolds. This was published in 1984. In 1984, I was 17. I'd been menstruating only for 1 year, menses having begun late due to a semi-starved body, one sculpted and carved by the conflicting resistances to and towards received femininities. The tinkering of body, food, and expenditure was an early bodily sculpture, and an unintentional nod to carving, a traditional sculpture made in 1972 by Ellen Anton, and the later homage by Castles and Cuts, a traditional sculpture in 2011 to 2013. I'm just presenting you with a temporal Mobius strip there. Pause. The artist Mary Magic performs a kind of hormonal hack. They articulate themselves in terms of body and gender as continually shifting in response to environmental toxicities, as they embrace unintentionalities and instabilities. One of the things they work with is estrogen. Pause. DIY emancipation of estrogen using domestic and lay scientific protocols towards more participatory distributions of estrogen. They ask, what is the feasibility of citizen science-based approach to synthesizing hormones? How can the recontextualization of laboratory biochemistry to an open source recipe outline the esoteric procedures and knowledges that are required to carry out such a process? Pause. They have run workshops in estrogen detection. Pause. Inquiring of the pervasiveness of estrocentricity. Pause. And this is also a quote. For estrogenicity is no longer limited to a small group of substances, but can be found in a whole series of chemical classes used daily in agriculture, industrial manufacturing, health, etc. Since the end of World War II, more than 10,000 active substances capable of estrogen activity have been released on the market and used in hydraulic or dielectric fluids for capacitators and transformers, glues, paints, detergents, insecticides, herbicides, fungicides, cosmetics, etc. Pause. Open quotes. We will ask multiple questions, says Mary Magic. For example, can we make our own hormones for contraception? What are the ethical implications in self-administering DIY hormones? How to detect the presence of hormones in the environment? How to design sensors that will play a role in citizen analysis of local water? Can we eliminate hormonal toxicity from the environment? End of quote. Pause. Eva Hayward describes the open potentials of the endocrine landscape created by our medications and industrial common materials, one in which our shared evolutionary history with other animals creates a common vulnerability to the effects of environments. She says, hormone levels change over an individual's lifetime and are affected by lifestyle, stress, physical activity, and exogenous hormones. Even natural plant substances like phytoestrogens interact with endocrine systems of various animals. Our material culture, as expressed by what objects we encircle ourselves with, the food we eat, the water we drink. Pause. Hayward invokes Bailey Kear's perspective in which he attends to the ecologically constitutive nature of bodies. He refers to bodies as constant processes, relations, adaptations, and metabolisms engaged in varying degrees of reproductive and economic relations with multiple other bodies, substances, and things. Pause. Astropause. Digging into the tinkering of muscles, its metabolic and hormonal sensitivities. Pause. I lift, pause. I breathe, pause. Other processes are at play at other scales, microbial, biochemical. Human bodies exert sweat, steam, and drip. Microbes proliferate on bodies and surfaces, finding nourishing niches in which to metabolize and expand. Weights rust, sweat clouds precipitate, seemingly immovable materials oxidize and react. Embedded in each of our human cells, symbiogenetic mitochondria, mosaic organelles of bacterial provenance from an origin evolutionary Eve metabolize. Muscle fibers fire and twitch in various tempos, slow, fast, intermediate. Satellite stem cells are signaled, differentiate, line up in multinuclear myotubes. Fibers are formed and muscle is laid. Pause. Between 2008 and 2010, I spent a fair bit of time in a laboratory in the University of Birmingham. I was there to research and experiment with growing muscle cells and spider silk. I worked with murine skeletal muscle cell lines and primary cell cultures, also from mice. I learned to observe and note their differentiation pathways, their minuscule architecting as satellite cells differentiated and formed multinuclear myotubes. I learned how to cultivate the cells, prodding them, nudging them in their tissue culture flasks and 98 well plates. Tiny muscular entities are cultured and excluded from any body and any body. Sometimes we would visit the animal facility where non-human animals are bred, housed and culled for research. A number of mice, specially bred, normally dystrophic, would be killed or sacrificed as quickly and efficiently as possible. I think humanely is the word that we use. Before tiny biopsies were extracted for cultivation. The lab was concerned with the tragic degenerative effects of Duchenne muscular dystrophy and with how to address this in human patients. It explored the molecular mechanisms of muscular dystrophy and investigating the genetics, hormones, and other cell signals that created its disruption of the differentiation lineages. The anarchic rationale that created exquisite-looking muscle cultures, spindle-like webs, clumps, and masses that refused the lineal logic of myotube construction. Cell cultivation and the many methods of inquiry take routine care and precision, regularity, observation, and adjustment. The research group was small and included one PhD student, Dean. Dean was a former bodybuilder who still trained. Every day he consumed from a series of Tupperware containers with unassuming regularity, chicken and rice and protein drinks, building and maintaining his muscle mass, architecting muscle on the growth scale whilst working with the delicate molecular biologic irrationale of dystrophic muscle. I could never establish if there was a connection for him between his muscle research work on the micro and his muscle cultivation on the macro. However, steadfast regularity was evident in both. The senior researcher, Janet, would show me how to peer at cells, how to sense their condition, how to be delicate and precise. In the dim tissue culture lab, amidst the pink flasks of muscle, she would tell me about long journeys she made by cross-country skiing. In her essay, Language of the Body, Kathy Acker writes, I want to shock my body into growth. I do not want to hurt it. I visualize and I count. I estimate weight. I count sets. I count repetitions. I count seconds between repetitions. I count ties, seconds or minutes between sets, from the beginning to the end of each workout. In order to maintain intensity, I just count. I put on Robert Ashley's 24-minute track, The Backyard, from his exquisite 7-part opera for television made between 1978 and 1983. My mind turns to my breath, 1. My mind watches my breath, 2. My mind turns and watches my breath, 3. My mind turns and faces my breath. 4. My mind faces my breath. 5. My mind studies my breath. 6. My mind sees every aspect of the beauty of my breath. 7. My mind watches my breath soothing itself. 8. My mind sees every part of my breath. 9. My breath is not indifferent to itself. 10. She never thinks of possibility or of how probably it is that they have come together. Those thoughts never enter her mind, nor do thoughts of sports. She has no desire to improve her muscles, for her piano playing is the only mystery. Thank you.
Thank you, Kira.
Wow. I mean, that was really incredible.
There's loads of chat.
There's lots of chat. We had a very productive chat session, which was also really, I'm really liking this Zoom thing where you can kind of collect and archive people's thoughts as you go. It's a real, like, awesome thing. So both of those presentations were resplendent in their own way, I think. And interesting to contrast their modalities and their kind of presencing. There's plenty of overlap. That's not an issue. I don't have to really strive to come up with that overlap. And even if I did, there'd be plenty in the comments to go to. So the moderation job has been made much easier by this chat archive. And I think what I'd like to do actually is ultimately to actually ask turn it over to some of the people in the chat who made comments to explicate some of those comments or to unpack them, as I've been saying throughout. But just prior to doing that, I actually just wanted to turn both of the presenters to each other in a way. And Kira, you mentioned when you were listening to Maggie's talk that there's a number of places you wanted to pause. By the way, I loved the insistence on the word pause throughout your piece of writing or your talk. A lot of people think we're really moved by that. But you said you wanted to pause at certain parts of Maggie's talk. And I wonder if you could recall that, like what you wanted to pause on and why you wanted to pause, or what you wanted to learn more about from Maggie's talk. And Maggie, I'm going to do the same to you to ask about your own impressions of Kira's talk. So just to turn the attention of the presenters to each other for a second, and then turn it out to the larger discussion group, which we have so much rich material in the chat to uncover. So yeah, so Kira, just to address that question, what did you want to pause on? Yeah.
Well, first, a big thank you to Maggie, because I think one of the things that enables a reading like mine is a presentation like yours. Normally these odd readings I do are within academic conferences where there's the maybe more traditional presentation of panels and academic papers and presentations. So the oddness of my reading, has that kind of place that it sits, that it's a sort of it's an unorthodox reading alongside other kinds of formal and very important traditions that are so important to how we understand and receive knowledge. And yours was done so beautifully and concisely. Gosh, I wish I'd written down more carefully the points that I would love to have expanded. One was definitely time, how I found myself kind of dwelling in this, in one hand, abstracted, but on the other hand, material generations, the F0, F1, F2, that containment, when you're describing that containment within the uterus of all these generations, it's almost like a sort of philosophical abiding or contemplation, as much as it is then how you invoke the epigenetic through different kinds of spaces, almost like this sort of and how dynamic it is. So that was one area where I just could have, well, I know those are two enormous areas in different parts of your talk, but I just could have hung out in those for a really long time. And just wondering for you, you know, when you're doing this work, I really appreciate, well, to some extent, that kind of the repetition, the precision that you have to have when you're creating these experiments, but also when you're performing them. And your acknowledgements at the end were really beautiful, and taught me something. Actually, I feel quite embarrassed that I haven't got an equivalent slide to you, when you're acknowledging people and the impact that they've had in helping you do all of this. But nevertheless, what you're doing with the material, I wondered about, you're working with all those material realities in the laboratory setting, but also there are these philosophical realities that you're working with at the same time. And also very, you know, when you began the talk as well with, you know, invoking and explaining to us and reminding us as well about toxicities and their impacts. You know, there's a kind of intensity to that too, in terms of thinking about people I know who've been affected by thalidomide, for example, what they're doing with that as well, particularly artists working with performance who are doing very, very interesting things with what it is to have non-normative bodies. And but going back to you, I wondered about, how do you dwell in some of those more philosophical areas, or how do those impact for you the spaces within your work, the ones that maybe you would put in the marginalia of a science paper if you could, or that might sit in the subtext, but that might not find a proper presence in scientific writing?
Yeah, so I think partially for me, like, to begin, a good example of, like, when you're talking about that F0, F1, F2, just like a quick example, is always the Dutch hunger winter, which happened during World War II when they cut off food to the Netherlands. Females were pregnant during that time, you know, nobody knew, but the Nazis cut off the food supply. So these females who were pregnant at different gestational periods went through some type of starvation. And this starvation actually caused differences in their gene expression with how they metabolize food in the F1 and the F2 generation. So that, I kind of always think about that when I'm thinking about that nested example, because that's a real life example. If you look up the Dutch hunger winter, there's, you know, popular science articles from the New York Times on it, and then there's research literature also on this experience. And then I guess for me, like, philosophically, it kind of makes me think more about my family history and kind of, you know, coming from both sides, like both of my parents, and what kind of thinking more about both of my grandmothers in that aspect when I do it. Because a lot of things, I guess you wouldn't necessarily think affect your health directly, but then when I'm thinking about this environment, it does get me more thinking about those aspects.
And for me, listening to science talks and not having a science background, it's often a funny mental exercise where there's an informational quality that comes across, but there's also like a parable that I'm trying to read into it in some ways. And the parable that I was looking for is something like what Kira was identifying, of this, what is carried forward. And if I understand the epigenomic principle well enough, it's that there's something that it affects a future individual that is not materially contained in the DNA or any transformation of the DNA. Well, how do you characterize that thing? Does it exist in space, or what is that thing, or how do you identify it or isolate it?
Yeah, it is kind of interesting, and I'm just recently getting into more so of epigenetics, so I would not say I'm an expert on this yet, but I'm becoming one. I think that the epigenome, you can kind of think of it like sitting above the genome, right? Epi, it's above the genome. It's acting as that regulatory network. So it does kind of just exist within the space of the cell, and there's different signals that are going to affect how, you know, there's different sciencey words, like we have different methyl donors, and then different proteins that are going to bring methyl groups to the DNA. So I kind of think of it as like the ghost sitting above above the genome, some type of ancestral something that's sitting above your genome and regulating how it's actually acting.
It's very interesting because the words you're using are these kind of meta, almost transcendent words, invisible, immaterial words.
Yeah.
But yet it has some kind of regulatory function in some ways.
Absolutely, yeah.
It's really interesting. I mean, I'm interested in what can be carried forward in a culture. Like, for example, if a trauma is felt, like say the dropping of an atomic bomb in Japan, and we're now losing the firsthand witnesses of that, but yet that trauma will be carried forward in the culture in some epigenomic way, perhaps?
Yeah, that's kind of what happened with the Dutch hunger winter, right? Like that starvation in utero carried forward to those grandchildren because they had been primed in a starvation-like environment.
And Tom, I'm addressing this to Tom Sherman. Is that what you meant by meta text data, or what did you mean by that comment?
Well, I was just interested in how, can you hear me okay?
Yep.
I got headphones on, so okay. I was just interested in how Kira was so carefully positioning her text, when in fact, I couldn't wait for her text. And then there
I
was
see.
And then there was so much information in the text that I got all the meta text information in the first few minutes of her speech.
Got it. Yeah, I actually enjoyed the preamble to that because there wasn't a lot of, there wasn't a big border for me between your preamble and the actual talk in terms of the pacing. So I enjoyed it. So Maggie, to turn the question back to you though, so hearing Kira's talk is such a different form of presentation, and yet there's some sort of commonalities, not just in the subject matter, but also in the kind of striving, the sort of effort in it. What kind of things were going on for you when you were listening to Kira's presentation? How do you relate it in any way to your own work?
Yeah, I think a lot of the idea of the sculpting and of just the two instances of like starting menstruation late because of starvation-like environments, and then also the male that you brought up eating like chicken and rice. I kind of really related to that because even as like a young female in quarantine eating a lot of snacks, I'm starting to think like, oh, just examples of that. And so I did resonate with some of those aspects of kind of what that's like, and then thinking of how that will translate into my future. And even thinking about, I told my mom a few years ago that she needed to start lifting weights. And so then when I heard that too, I was kind of like, oh, it just made me kind of reflect more about my life, like in my young 20s, and then with those people around me, and kind of how to, I don't know, how I'm growing and changing now, but then how also I will in the future as well, kind of. I guess, and relating to my own work, I think at first I was really just listening, so I wasn't quite relating it back to my own work a little bit, but I also just think like the really careful putting together of the work and how everything flowed in such a way that the story like changed and evolved and took many directions, but really went together. And there, again, has to be a lot of pre-planning in that.
And Maggie, what motivated you to participate in a bio-art mixer? Do you do art of any sort?
I don't really. No, not at all. I really like learning about other types of work, and I really like sharing my science with other people and making it so that it's kind of translational so people can understand it. If anybody asks me like, what are you researching on? I'm like, oh, thanks. I've been dying to tell you about it. So I think like having a space to share that as well, where people want to listen. And then it's just really cool being able to relate, the sciences to the arts also, because I think that the communication isn't always there.
And Kira, you were talking, or just mentioned earlier a second ago, that talks like Maggie make your talks like yours possible. Did you mean that just in the sense of they provide a forum for it, or do you mean something like it's the foundation upon which your investigations are based, or how did you mean that?
Well, maybe a bit of both, but I think because normally I'm doing these kind of readings in academic conferences where there'll be a convention of, presenting academic papers. And so these readings I do are trying to invite or trying to intervene into the academic conference setting somehow. So it's really helpful. I think it works better when it's got when it sits alongside other genres of writing and presentation, like research papers and scientific papers, but also because it is so because my thinking owes so much to researchers that I've been around and spent time with, including in the life sciences. So I think a lot of my thinking has been, and the sort of the huge shifts, the monumental shifts that I've gone through in my artistic thinking and doing and making, does owe a huge amount to those conversations I've been able to enjoy with researchers in science for the last couple of decades. Very much.
There's a couple of comments just coming in at the bottom about how academic conference need more of this. Caitlin, I think you made that comment. What is this? What do they need more of in your?
Transdisciplinary work, I would say. So work that, because it is, and I'm only familiar with science academic conferences, so I can't say if humanities academic conferences maybe have a lot more of this, and I think even science and technology studies conferences have a lot more of this, but the liberatory potential of stepping into new media, I think, has been very much underexplored in scientific conferences.
Yeah, and it's always when faced with this proposition, I always feel that as someone in the humanities, that I'm going to need to somehow convince scientists that this is needed. I'll have to make my argument in some way, but maybe not. Maybe it's just already agreed that there is this need or desire to interface these two techniques. I guess what you would come up against potentially is like, how is what Kira did knowledge, and do you ever, and does that conversation ever come up for you, Kira? I believe it's knowledge. I believe that art is knowledge in some way, but yeah, just to throw that.
Yeah, I mean, I think for any of us who spend any time in academia, there's this ongoing thing of how do you legitimize artistic methods within academia? Some people will throw up their arms fabulously and say, well, why should one try and conform to this idea of knowledge or production of knowledge or those kind of politics and those drivers and those economics of production of knowledge? But I think where my kind of leaning is in this idea of knowledge and experience, that there's definitely, I haven't done a residency for a while in a science context, but whenever I have, it's tended to be for a while, particularly when I was in Birmingham. And what I would tend to find is more similarities than differences. And I'm not saying art and science is the same. That's not what I mean at all. I mean through the kind of practicing with ideas, practicing with emotions, practicing with affects, and that those coalesce into all kinds of experiences and ideas that many of us find sympathies and synergies and empathies with. So I'm not sure what I'm doing, if what I'm doing is positioning knowledge per se. It's normally trying to open up a short amount of space and time in which to invite some kind of shared contemplation on something, and that's normally because there is some kind of action within that, some kind of process, some kind of experiment of performance or performativity within that. But of course one can't control what then the outcomes of that are. They're going to be varied depending on people's individual reception. Kind of answered that question quite badly. It's a huge answer.
No, it's a big question, and I do feel like I wish I had a better vocabulary with which to express how art can be salubrious in these types of academic conferences or just within the academy. But so maybe a related trajectory here. I want to take another question in the chat. The most recent one. I don't want to be subservient to the tyranny of the recent, but the most recent comment here is, I'd be interested to hear Maggie and Kira discuss the relationship between systems and identity. I think there's a lot in that question that also relates back to some of the earlier comments. So Maggie, do you want to take that first, or? Oh, you're muted.
Sorry.
Yeah.
Can I like clarify systems like biological systems?
Yeah,
And
well,
I,
actually, yeah. Ann
like-
Osborne, do you want to explicate that, or do you want to set that up in some way? Let's hear from you. Where is Ann Osborne?
Oh, sorry, folks.
Hey.
Hi. Well, I think that I'm interested. So my work tends to be I'm a media studies person, and so I don't tend to think in biological systems
okay.
generally. But I guess I'm curious in how, and I think it does relate a lot to what Kira was talking about as well. I'm doing work right now looking at how media discuss transgender individuals. And so I'm interested in how systems which include biological systems limit our sense of identity, and how we often, the sort of push and pull that exists there. So I'm not sure if that clarifies.
Yeah, it does. I think for me personally, and even putting this presentation together, I just noted certain aspects and things I'm going to change probably for my defense. For F0 generation, like, I always say mother, kind of. When I looked at other literature and aspects, that's, the F0 generation, the gestating mother. When in reality, I could just say gestating individual, because it can be an individual who has ovaries and is carrying a fetus. It doesn't have to be somebody who identifies as a female or identifies as a mother. And I believe all of these things, and these are things I take into my daily life. And then for me personally, sometimes when I get into the scientific setting, I think I fall back on the literature or on what I've seen more so. So I might be more apt to, and I did write gestating mother. So I think for science, that these are ideas that I need to start inner working in my work, especially because I work with reproductive biology and I'm talking about these systems, and they're truly just reproductive organs that an individual has. So I think for me, kind of taking away that colloquial mother, father type of thing and maybe focus more on the reproductive organs. I don't know if that answered some of it.
Yes, thank you.
That was a great reply, Maggie. I'm just reading the question again. It's such a good question. I think when I was first making performance work in the sort of late 90s, early 2000s, the kind of work I was making was a lot more concerned with identity, I think, and was very much, not that it's not now, but that was the kind of deliberate emphasis, I think, at the time. And then as I had all these kind of dissolves, as I was, entering into working with biology a lot more and biotechnology, I started trying to, realizing that I needed to find other kinds of languages and other kinds of ideas to understand these huge shifts. So in the late 90s, early 2000s, I was really interested in the much more sort of psychoanalytic construct of the knotted subject, in which multiple narratives and power relations would operate within one human body, and would construct that sense of subjectivity in different ways. God, I'm trying to think whose essay I'm sort of half quoting there on the knotted subjectivity. I can visualize the book, I can visualize the essay, and the author has gone from my mind. It might have been Griselda Pollock or someone like that.
We have quite a robust list of references in the chat, which I think I'm eager to capture. We have some good research being done in it.
Let's not forget the chat save function at the end of this. I'm going to be really curious to look at that. But I was, where am I going with that? And then I think as the category of human, became more, as I became more suspicious of it, I suppose that's where I began to understand systems more. But what I find that I do is this is where I invoke the sort of multiple perspective. to have a sort of multiple perspective at any one time, not to have a singular one where notions of identity appear and disappear. I was listening to Patricia McCormack earlier today, who's an academic based in the UK. She's just published her book, The A Human Manifesto. And she's also, in the context of the interview, she was also talking about her occult practice. And she said something I thought that I really felt I understood, which was that when one works with ritual, the first thing one has to give up is a notion of the self, or a self, or oneself, or something that has continuity, that there is a continuous self. And then later on, when she was talking about the A Human Manifesto and her critiques and interest in the ideas around the Anthropocene, she was talking about this loss of self, this necessary demand for loss of self in this acknowledgement of human exceptionalism, and also as a way to move away from human exceptionalism. And I thought that was a really interesting point that I felt I could relate to in my way, that there's this kind of appearance and disappearance of self and selves. But then the important, for me, point to pause at here is always whose identity, and whose identity needs to be protected, and whose identities then become obliterated or marginalized or forgotten or buried, or so it's also this complicated thing of from which perspective am I speaking and thinking, and what are the material operations of that, what are the real life effects and conditions of that. So hence needing to sort of come back to the sense of multiple perspectives.
This would be a good moment. I think there's been a lot of activity in the chat comments about. Oh, I'm sorry. Please, yeah. Before I move on,
I was just gonna say, I think going back, this is a great example of why it's great to have talks like Kira's at scientific conferences, just like we were just saying, because it also makes me think about my work differently. It takes me kind of out of the primary literature and out of the data of coding my methylation, kind of thinking about why I'm doing the work, and then also who the work relates to. So I think it's just, it opens up my mind again about the work. So
that's great testimony. Yeah, it's it has the effect of de-instrumentalizing or deprofessionalizing. No, that's not quite the right word, but always with science, I'm always worried about the capture to science with the big S, and that whole discourse around how it's funded, how it's monetized, how it's utilized, and so on. And you feel like there's something about this discourse that recaptures the idea of like pursuit of knowledge or pursuit of understanding, let's say. But I wanted to, it felt like the conversation was moving in a good direction towards a topic that was brought up frequently in the chat around transgender issues, around gender issues in general. And I wanted to, maybe if I could invite Kathy High to interject here. A lot of the initial comments were from you, Kathy, like sort of what was your perspective, or what do you want to just sort of summarize some of your comments, or pose a question from some of your comments?
Hello. Yeah. So I'm
Whoa.
taking a still frame from Kira, because Kira, what I loved were the glitches that came up in your, I can't even do it. The glitches that came up in your talk when you kind of broke the green screen, which made me so happy. And it was so incredible and like just magic. So, and both of the talks, Maggie too, super magic. I'm super stoked. And I don't know, I've got so many ideas running through my head at this moment. It's hard to kind of come up with talk, you know, specific questions, but because I had big questions as we began about deep time, and I think Maggie, you addressed these so beautifully, and you too, Kira. So I super appreciate that. I mentioned something in the chat about crypt time, and then somebody else, I think it was Caitlin, maybe came back with kind of queer time. And these are concepts that come up in disability studies and queer studies, rethinking our approach to time, which of course all of us have to do right now at this very moment. And it's so unusual and so interesting for all of us in some ways, even though it's an incredibly challenging moment, I know. In terms of queer studies, I'm teaching a course right now on queer ecology, and it's the first time I've taught this course, so it's clunky, but it's really looking at, and clunky in that it needs to be reworked all the time with the people, the students and everything, but it's been a joyous semester. And many of the things we're looking at is these kinds of definitions of gender, of our kind of essentialism around thoughts of hormones and things like that. So I do appreciate the way both of you are thinking through the questions of this. I was really touched, Maggie, by your comments of readjusting the word mother in your presentations. And Kira, of course, I super appreciate the ways you're thinking through menopause as a more of a kind of environmental issue. In the class, we've been looking at things like Annie Sprinkle and Beth Stevens' ecosexual approach to, applying sexuality to ecologies, and what does that even mean? And I think that these kinds of ways of bringing interdisciplinary approaches to all kinds of disciplines, while it can be very messy and sometimes kind of very too vague or broad, at the same time it allows us to make these other kinds of connections that I see absolutely happening tonight. So I don't know that I actually have an absolute question for you guys. I just want to say that I'm super stimulated by what you've been talking about, and thank you both for this. And thank you, Ed and Kira and Maggie and Boreana and Heidi, for making this whole thing happen.
I really appreciate your capturing the screen and putting it up there. Kira, I think when you review the video of this, you'll be quite happy with the way that the background image functioned in your talk and the way you would disappear into it and then reemerge from it. A number of people noted that kind of liminality or disappearance of the self, or the sort of articulation of permeability that the back image represented. So
I can go disappear into Kathy. Disappear.
Oh, whoa. That's amazing. That's really great. So there's a couple recent questions here in the chat from Julia. So you want to present your question, Julia? Julia Zay.
Sure. I was just wondering, partially in hearing Maggie talking about, whether to refer to the individual in talking about the mouse, and also with the discussions that have been coming up around humanity to some degree, what all of this means in using the model animals in the lab, and how you're referring to these individuals and the relationship between the human and the animal model in that sense. I thought that would be something interesting to hear the presenters' thoughts on.
So thinking about how I think about my mice in the terms as well, to be completely honest, I don't really think I've thought about it in that sense yet, and so this is also why this is beneficial. Typically I have my mice named on like a BT 101, behavior testing 101, and then just very scientifically numbered and ear clipped. But I also think that, like I said before, going back, I can kind of more, I don't really know. This is a hard question, Julia. But I think that partially I can still go back and think about how I use the model system. And again, like just gestating mouse instead of, again, referring to as mother. Because I always refer to the zygote or the fetus just as that. I don't ever really say baby. So then it's kind of interesting why I carry over mother to mouse, and I leave like the fetus as that. I kind of keep that much more scientific. So I guess that's something that I kind of just thought of. I don't know if that answered your question.
Did that answer your question, Julia?
Yeah, that's great. Thank you, Maggie.
Is there anything you wanted to add to that, Kira?
I was thinking about how writers like Ida Benke are doing beautiful work. She's a academic and a curator based in Denmark, I believe. And she is part of the Laboratory for Aesthetics and Ecology. And she's been doing some really beautiful curation and writing around the category of mother and other, and playing with the whole sort of spelling of that, you know, the mothering, the othering of mothering, and so on. And I think her writing is really, and her curating is really beautiful to and important to look at in terms of how we can examine that word and not always abandon it, but not allow it to be so, but allow it to be expansive so that it can include many other kinds of bodies and identities and people who wish to deploy it, use it, be embraced by or embrace. So she writes sort of across multi-species others as well. She wrote a bit, and I know her writing well because I had the pleasure of co-editing a book that we invited her to write an essay for, precisely about this, a book called Art as We Don't Know It. And there's a free PDF of it, and it's I just think I think that kind of writing and that kind of thinking and curating really adds to this kind of thinking and these decisions. I think to have, it's also so important to think about where words are as well. The other thing I wanted to say is that something that I haven't written about yet, but I hope to co-write with someone, is around transgender experience of menopause as well, and just also the idea that there's cross conversations that should, could, and ideally would be happening around menopause, trans women, particularly around hormone replacement therapies, gender confirming therapies, medical research and neglect of medical research into those for trans people, but also what is it to sort of uncategorize menopause as well. So these are conversations and inquiries that I'm hoping to have. Yeah, that's something I've been thinking about a lot.
Caitlin, can I call you into this conversation a bit? You made a number of comments. First of all, thank you for providing so many references throughout the chat. We will save the chat and those will be preserved. But you made a number of comments throughout the chat about liminality that seemed like they might carry on from some of these comments by Kira and Maggie. Did you have anything you want to say or add to this discussion?
I think the thing that I most need to say first is I've had some really wonderful conversations with an early career scientist. She's a postdoc, May Guthman, who's a trans neurobiologist who studies what hormones do to the brain and does a lot of work. And we're working on work together about the way that, like, assumptions about hormones and that, you know, estrogen is a women's hormone and testosterone is a male hormone have biased us not to understand the fullness of their responsibilities across organisms, and the ways that we expect to see sex differences then reifies in our work, seeing those differences in the way that we even design our studies. And so the ways that we build in, and this is I can't remember the work of the philosopher of science who did it now, but that the ways that we ask questions have a predisposition for the answers we're even willing to accept, and seeing that in kind of being broken down in the work that we talked about today.
Kathy says, is that Bruno Latour? Could be.
No, it's not. It's
nope.
It's a woman.
Okay.
Elizabeth
Good.
something.
Good. Well, do you want to respond to that, Kira or Maggie, anyway?
Sorry, I was just thinking.
Well,
about,
I distracted everybody with my Bruno Latour interjection.
no, it's all right. It's just for a moment I thought, oh, Elizabeth, who is that? I had a thought in my head that just didn't properly announce itself. No, I just think I just would like to know more. That's my response to that.
Mm-hmm.
I just thought, oh, I want to know more about this research. That sounds really incredible. But also I think that really important point about how a question will already anticipate or have nested or niched within a desired, a sort of systematic outcome or something that already fits within that system or that avenue or that construction. I think that's what you were saying, isn't it? Which I thought was really important.
Yeah, I have thought a lot about the naming process throughout both of these talks and how naming is active and has a sort of, it's a double-edged sword, but also inevitable. I wanted to go back to an earlier moment in the chat, just to there's so much good activity in the chat to get as many people involved as possible. Melissa, you've been pretty active throughout, and there was a couple moments where you kind of called out a phrase, and I wanted to unpack it a little bit. And one of those was environmental toxicity. Can you explain why you pointed that out and what's on your mind with that?
Can you hear me?
Yeah.
If I can remember why I said about toxins, and then Kira was talking about environmental toxicity related to to menopause and to, I think that's what you were saying, Kira, right? And it just struck me how much more related the two talks were than I ever thought they would be. So.
Yeah, me too. They really were actually quite related.
And then Kira also mentioned different estrogen, which is one of my favorite molecules, and different estrogen mimicking compounds. And that's that's not what Maggie studied, but that's one of the things we study in my lab. And it just I just loved hearing about that. And it helped me to think, as Maggie was saying, it helped me to think about what I do in a very different way and appreciate it from a very different view. So I thought that was cool.
Great. How did it make you think about what you do differently? Can you articulate that?
Well, I just mean, I'm just like a very narrow-minded scientist, and I don't think about how, like transgender issues. I don't think about how my work might relate to something like that,
Mm-hmm.
or that it even relates like in a more artistic way, and
yeah.
There was some. Sorry, I lost my train of thought.
No, that's okay. I just wanted to hear from you on that. Yeah, and I think were you the one pointing out something about mimicry? Because I also was thinking about mimicry and this idea of like there being a copy, like there's a copy of the genome from one to another, but there's some influence on top of it that then changes that. Is it trying to copy that? Caitlin also was calling out the idea of mimicry.
I don't think I talked about mimicry. I can't remember who did, but.
I think, Melissa, you mentioned how DES mimics estrogen.
Oh, I did. Oh, yeah. Yeah, because there's different compounds in the environment that mimic estrogen. Compounds from plants. There's also synthetic compounds, and then I think in Kira's reading, she was talking about like different, like cosmetics, and I can't remember what else, but it just related so much to one of the aspects of what we study in my lab, and in such a different way. It was just very cool.
Cool. The other thing I wanted to call out in the discussion was Laurel. At one point you mentioned or called attention to this idea of inhabit. I'm trying to find it in the chat. Laurel, are you still with us? Do you want to talk about inhabiting and habitation?
Yeah, I don't remember exactly how I phrased it, but I think, I mean, in regard to the embryos, and I mean, they inhabit their space and then they grow and morph and become this being. And then, I mean, just from the concept of our own personal identities, within what Kira was talking about, our identities are taken from everything about us, and whenever that changes, it shifts and changes with us, right, with our identity. But I feel like identity is a permanent thing, and our being maybe is not so permanent in some bizarre way. I don't know why. Identity it carries through generations, you know, obviously with some kind of markers that can be put from, you know, embryonic stages. I don't know. One other little thing, I went to the Cruel April. I listened to the Cruel April reading, which was just last night. It's at Point of Contact down there. And it was Erica Martinez. And the last line of the first poem that she read was something about, you know, having an existential crisis when you cut your fingernails, right? Because we're changed by everything that we do. And inhabiting ourselves it's like a embryonic moving and growing, I feel. I don't know if I'm making any sense. I'm just getting all weird and having a drink here. It's great.
Sure, that's what we're supposed to do. Like, I think we should be very visible. Don't hide your drinks. They're
totally doing
they're
it.
happening.
Friday night, we're winning, people.
That's right, we're winning. Flattening the curves.
I forgot about the drinks bit.
Inhabiting my space here. Yes, I am. I own it.
Yeah, I've been a little bit too inhabiting my space, I think.
I'm enjoying this very much. This is great. Yeah. Thank you both.
Me too. Any responses to that, or from Kira or Maggie, or you riffing on that? I wanted to go to a slightly different direction. It's almost 9 o'clock, so I probably feel like we should allow people to start leaving as they want. You can sneak away. You know, there's no rules here. A lot of people spend a lot of time on Zoom, so if you're Zoom fatigued, feel free to exit. I feel like I have to say, Kira, something about the repetition of the word pause, which was a very effective technique, I feel like, in your reading. And by insisting on that pause, first of all, it gets us to pause and refocus, but it also does something to articulate a notion that there is some sort of flow of time that's ineluctable, that you can't escape, but then you escape for a second, but then you must return to it in some way. Or that's what I read from it, something like that. I wonder if you could comment on the device of repeating the word pause, or what pause means as it's separated from menopause. Talk about pause, I guess.
Yeah, well, you know, sometimes when I write things out that I've got to read, I give myself little reading directions like pause. Just tell myself to pause, or tell myself to say something louder, or whatever it is. And I think it, you know, originates with this riff on menopause, but also, of course, wanting to perform time, which we're doing all the time, but just wanting to make that explicit, I suppose. So it does become this, like you're saying, it's an interruption, but it's also just brings back into the attentiveness to time, and that we're sharing time together as well, that it's me reading to you and acknowledging that we're in this little special moment of time. We're not just, you know, there are numerous temporalities I'm trying to acknowledge, including the one that we're in right now, this straightforward one.
This pause.
This
Yeah.
pause.
Yeah,
so,
this pause.
It's also a kind of a cheesy reading out loud to people trick as well, you know, especially when there's, I think when you're in the same room as a bunch of people, and you can really play with that idea of I'm taking a pause and I'm saying the word pause as well.
Yeah, it comes across as both an instruction to yourself and an instruction to us. And I think that's very effective.
Yeah, and it can be a little bit humorous, a little bit of a wink of, you know, here we are and I'm reading to you and I'm trying to pace myself. And then, you know, if there's a microphone right in front of my mouth, then I can do the sighs as these big sighs into the mic that people hear in minutia. So it's those little reading tricks, I guess. But I had no idea how that might work on Zoom.
I must say I found the entire talk very effective. I mean, your reading I was very moved, and it was very effective.
It's nice to have the opportunity to try this out in this context with Maggie. So big thank you.
So it is 9 o'clock. I know that I myself have to actually depart. I have a child that I need to put to bed and so on and so forth. I've really enjoyed this, Maggie and Kira. Thank you so much. I need to depart. Boriana, do we want to formally close this, or do you want to keep it open, or how do we want to do it?
I think keeping it open is not going to contribute more to personal conversations, because they kind of will have to happen publicly on this Zoom. So maybe we can close it, and I really hope, and it seems to be the case, that some of the conversations will continue, or new conversations will occur in other places, in labs or in studios, in art studios, that will be related to this conversation today between the two presenters and among us. I also think that it was surprisingly, I'm missing the word in English. It's not effectively collaborative or overlapping, but kind of connected. The two talks were connected in a very interesting way and amazing way at the same time. I was not participating in the chat until now because I wasn't able to figure out how to get into the public chat, and I had this private chat with Kathy Hai for some reason, and I didn't want to talk to her. I wanted to talk to everyone, but anyway, I just didn't participate. I wanted to mention something at the end that, especially because you were talking about the pause, the word pause. There is this performance by Faith Wilding, which is Waiting. Kira, maybe you know about these performances made in the 70s, I think, or the late 60s. And it's waiting for me to start to menstruate, waiting for this, waiting for that. It's waiting. So it's a lot of waiting, the repetition of the word waiting. And I'm thinking that there are also this performance and your talk performance, your reading performance now, were connected and extended. They kind of live in a similar space, but it is so much more extended into much more areas. Your reading is much more extended into these areas, although it has this repetitional, this rhythm that seems, it's actually very different. If I remember the piece, it is much more repetitive, but still I was just thinking that there is this connection between these two works as well. And yeah, of course, I wanted to thank to you, Kira, and to you, Maggie, for the amazing presentations and for the conversation that has happened now. And I really hope that it has been effective for everybody else. Thank you so much.
Thank you. Boriana, are you going to share? Are you going to save the chat?
I have saved it several times because I usually lose things. So I probably have like five files with it. But yeah, I just saved it again. I'm recording also. I think I'm still recording, and that's we don't know if the recording will be going on YouTube yet, but I have it as an archive.
Great.
Or how public the recording will be, we still don't know. It will be something that we'll decide later.
Wonderful. Well, good night, everybody, and thank you.
Right.
Yeah.
Thank you
so much.
Thank you.
Thanks.
Oh, okay.
Thank you.